Welcome to Biotech Stage. It's a stage where we meet with different biotech professionals from the globe to discuss something very exciting, something very hot, and something very informative about the biotechnology world. So today we have Harshit Shah. Harshit has been working at Dr. Reddy's lab, heading the MSAT group. He has worked at different companies in past 10 to 15 years, including Zydus, Cadila, Intas, Hetero. So welcome Harshit on this show.
Thank you.
So, Harshit, there are a lot of things that I want to discuss with you. You have been in the industry for almost 10 to 15 years, and Indian biotechnology industry has grown a lot in past of 10, 15 years. I still remember when for the biosimilars or MAB, use of serum-free media was a new thing. Use of protein-free media, use of animal component-free media was a new thing, and now it is fully developed. So you must have seen all the changes. You must have seen different obstacles. You must have faced a lot of challenges, and you must have overcome all those problems. So if you could please share your journey in the biotechnology for last decade or so, that would be great.
Yeah. So when I started my career, I know that, as you mentioned very correctly, that we are thriving towards the proper media, proper synthetic media, and animal-free media, and those kind of complexities which we are facing earlier. If you see in last 5 to 10 years, how the industry is shifting, that nobody is talking about the media, serum-free media, or animal origin-free media, or chemically defined media. Now the transition happened from the low cell density to high cell density kind of approaches, process intensification approaches. So we can see a very different kind of dogma now, and shifting from the conventional thinking towards the intensified thinking of the processes. So I know
Mhm.
when I used to do the process development during my initial phase of career, people used to afraid of using the TFF membranes, and they are thinking that the protein will get aggregate. Now,
Mhm.
if you see itself in the tangential flow filtration, there is a huge shift, like from the very small scale to very huge scale. I have used 100 meter square TFF system. It's like the entire room kind of a thing. And other than that, now the technology wise, there is a single pass TFF systems are coming, and with that we are also focusing on the high concentration formulation through those TFF systems. So we can understand that how it is a shift in the thinking, in the understanding, in the exposure, and in the execution. Now people are talking about the intensified processes, where we are using the different kind of components which are additives like vitamins, minerals, lipids. All those things are also in a such a way that we will grow our cells in a huge quantity. We will produce a huge
Mhm.
amount of a protein, and that will also make the life very easy for the biotech organizations in terms of the cost competitiveness, and you can say cost erosion. Now,
Okay. Okay.
considering the challenges, so challenges
Mhm.
wise, I can tell you the challenges is when we used to do this kind of a optimization. We
Mhm.
don't have enough technologies at that time. We used
Mhm.
to run normal techniques, then it will take some times to analyze, put that in the systems, taking the data, again analysis. Now, those challenges are minimal in terms of the data. We have very high throughput screening systems. Earlier, we used to do 30, 40 experiments to have a process developed for the from the vial to the vial, like from a vial thaw to the DP vials. Now, with the high
Mhm.
throughput screening within a month, we can do within the 10, 15 experiments, we can screen our processes, we can define our initial processes, and we can go with a very faster in terms of the product lifecycle management from the initial stages, or you can say early stage of development to the late stage of the development.
Okay. So you mentioned low density. It used to be low density, and now there is a focus on high cell density. So obviously, that will give more protein on a per cell basis or per liter basis. That will cut down the cost.
Yeah, yeah. So earlier is like, as we know, that's a 1 gram per liter. Now with
Mm-hmm.
the now people are talking about 10 gram per liter, 15 gram per liter, 20 gram per liter, and through the
Mm-hmm.
continuous manufacturing processes. Now continuous manufacturing also came into picture. So people started now talking about the $70, $50, $40 per grams of the MABs. In that particular scenario, now it's evolution of the entire biotech thinking
Mhm.
and the strategies are coming into the picture.
Okay. So when you say continuous manufacturing, so it is not fed batch focused manufacturing, it is more on perfusion and continuous manufacturing.
Yeah, yeah. So earlier is like batch mode. Then
Mm-hmm.
in the 2 to 3 years, it's shifted to fed batch. After
Mm-hmm.
that fed batch, it shifted to the intensified processes like perfusion, but with the concentrated fed batch. And now it is coming like the continuous manufacturing. So there are the
Continuous
four
manufacturing.
different eras of the bioprocessing are being seen by me during my entire career till now.
Okay. It is amazing to see how the biotechnology or the biosimilar or novel maps production and the process development has evolved in past 15, 20 years. And instead of 1 gram per liter or even less than 1 gram was used to be okay, but now in now it is a tenfold increase, about 10 grams per liter. So where do you think it will be in next 5 years or so? I mean, what are the new things coming up?
Yeah. So next 5 years, in terms of the molecules, so molecule wise, you will see lot
Hmm.
of bispecific antibodies. You will see lot of
Okay.
trispecific antibodies. You will see lot of ADCs, so antibody drug conjugates, and you will see lot of cell and gene therapy products also. So if I can say like that, when I started my career, I can say one or two maps names like adalimab or rituximab and those kind of maps. Now there are almost 100 molecules are there in the research and in the clinical phase and in the late stage of the commercialization phase. There we can see the hundred of different monoclonal antibody itself with the different indications for the patient's use. So
Hmm.
that are coming. Other than that,
Hmm.
second, that all the entire biotech industries are going towards the intensifications in terms of the they want a speed to the market, they want a multiple products in the pipeline for the research and then clinical phase to the commercialization, and they want very faster regulatory approvals also. So there is
Hmm.
a
Hmm.
shift in the regulatory landscape also, so that also we are experiencing now.
Yeah, thank you so much for naming all these new things that are coming up. Like you mentioned, bispecific, trispecific, ADC, and cell and gene therapy, and intensification, speed to the market, and multiple products in the pipeline. Those are the key for success for the companies as well as for the patients. Because if companies are going to have more products in the pipeline, obviously there are more chances of some of those products will become actual commercialized molecule. Rashid, one other thing which I wanted to find out from you is there are a lot of new techniques came into picture, and there are a lot of things which are sometime you work on the bench and or you supervise a group working on the bench. Sometime you think like, oh, I wish I could have that. So do you think you can name one or two things that you wish should be available now and they are not in the market at this time, whether it is in terms of any assay related thing or any process related thing, anything that you could name at this time?
Yeah, so I want to focus two things here. One is the
Mm-hmm.
bioprocessing technologies. So
Mm-hmm.
let's say, for example, currently MAPS purification
Mm-hmm.
needs a good membranes, especially in the protein A affinity chromatography steps.
Mm-hmm.
So protein A affinity chromatography steps is reducing the burden of entire purification, especially for the MAPS, for the fusion proteins, for the bispecific antibodies or trispecific antibodies. So especially antibodies needs that kind of a membranes, because as we are seeing that there is a evolution in the regulatory landscape, especially from the US FDA and other higher
Mm-hmm.
regulated markets like European agencies, US FDAs. Those are expanding their regulatory landscape. Now,
Mm-hmm.
considering the balancing the innovations for the regulatory landscape, this is a very good innovation that can balances the regulatory landscape, because that will cut down lot of other points or considerations or concerns from the regulatory authorities linked to the conventional columns, packings, its
Mm-hmm.
reusage, its storage,
Mm-hmm.
its microbial control strategies. So, considering all those points, it is very important for any of the biotech service providers, or you can say drug manufacturers, that those kind of a membrane technology, especially for the protein A chromatograph, is not, you can say multiple players are there with the very
Mm-hmm.
good intensified processes, needs those kind of a membranes to process the protein purification very faster manner and efficient manner.
Mm-hmm.
So those technologies
Okay.
should be easily available in the near
Okay.
future. Second, I want
Okay.
to talk about process analytical tools. Now, as we are growing 15 grams per liter, 20 grams per liter protein with the very high cell density cell cultures, especially for the CHO cell lines. So, CHO
Mm-hmm.
cell lines, when you're growing with the very high cell densities, there is a physico-chemical liabilities are associated with the protein molecules. And
Mm-hmm.
if we have a good process analytical tools with AI and machine learning, if we integrate this particular three concepts, that can
Mm-hmm.
help us to take a decision making to develop a process efficiently, and in the commercial space it will help us to track and monitor the process without any major waiting time for the analysis and then data analysis, and then you can take a decision. So just to
Mm-hmm.
cut down that conventional process testing, in process testing, instead of that we want to focus to the real time release testing. So real time release
Mm-hmm.
testing is nothing but it will be possible through the process analytical tools, and that also
Mm-hmm.
there is a very less players are there, and technologies are also not that easily accessible to the
Mm-hmm.
manufacturer. So that also I want to emphasize on, that it should also be available with the good accessibility.
Okay. So hopefully these two things will be solved in coming few years, and those technology will be available for use. Okay. So Harshit, one other thing that I wanted to ask is, which I think everybody is talking about nowadays, and that is artificial intelligence. AI part is affecting almost every single industry. So, what do you think AI will change in biotechnology, and how this could be useful? Or, I mean, maybe there are some cases where it cannot be useful. So, if you could please say something about artificial intelligence in biotechnology.
Yes, definitely. So, artificial intelligence, also there is a evaluation framework is there. Earlier, when I used to face US FDA or audits or inspections during my 2016-17 means year, so that 2016-2017, I used to use like T test and the equivalence test to prove the comparability. But
Mm-hmm.
now, considering the artificial intelligence, as I told you, that if I will integrate the artificial intelligence in my processing, I can upfrontly prepare a golden tunnel kind of a concept. That golden tunnel will help us to monitor a process, because if you see, there is a huge cost
Mm-hmm.
involved during the scale up and right first time scale up and the manufacturing of the different molecules with the different molecular natures. So for that we need a artificial intelligence that it will minimize the time from
Mm-hmm.
data analysis to decision making. So that is very
Mm-hmm.
important for the artificial intelligence. Second, how the artificial intelligence will help us as a drug manufacturer or drug designer, basically, that
Mm-hmm.
through the artificial intelligence we can focus on the good drug interactions, we can predict the interactions into the patients and animals, and second we can engineer our biotech products, especially we are working on the protein molecules. So what happens
Mm-hmm.
if you use the AI to go for a better engineering of the molecules? So that can also help us to reduce the problems which are associated with the physicochemical liabilities of the protein chemistry. So that way also AI is good in terms of usefulness. Only that how AI we should use is that when we are saying that our data acquisition for the
Mm-hmm.
artificial intelligence, it is very important that our data is very accurate and it will be acquired immediately. So where
Okay.
we are saying like some of the concepts where the data accuracy may not be that great, there we should a little bit careful about the usage of the artificial intelligence.
Oh, okay. And you think this will be available in coming few years, 1 or 2 years, or it will take a little longer?
I will say it is available, but accessibility is less. And second,
Mm-hmm.
especially for the Indian drug manufacturers, they need to shift their mind. Currently, their mind is drifting from conventional modes of the operations to the artificial intelligence, but they need to
Mm-hmm.
shift their entire focus
Mm-hmm.
from the conventional things to the artificial intelligence, because sometimes what will happen that we are too much reluctant that without that also my things are working, but
Mm-hmm.
instead of focusing on the only just working, we need to focus on the operational excellence of those particular unit operations. So that operational excellence, we need to shift our mindsets, especially
Mm-hmm.
from the Indian manufacturers, from the conventional to the artificial intelligence, that we need to focus on. If we shift our focus, then in 2 to 3 years, it will be very easily we can say that the artificial intelligence can be useful for the upfront decision making for the our bioprocessing. Otherwise, we that shift is little bit slower or steeper, the slope of that shift, then it will be almost, it will take around 5 years.
Okay. So hopefully, the Indian biotech industry senior management would be watching our podcast and listening, and I'm sure they will be thinking about it. They are already thinking about it, and they will, as you mentioned, will switch towards full focus on artificial intelligence. So another thing, which, as I mentioned before the podcast, that this podcast is geared towards a younger generation, people who have just finished their PhD or masters and entering into the biotechnology field, or have maybe 2 years 5 years of research experience. So what advice you think you want to give to them, how they can excel in their career, what they need to focus on, what they need to little bit avoid, and how they can get into this biotechnology field in a more successful way.
Yeah. So first and foremost thing that any young person who wants to have a career in the biotech industry, they should
Mm-hmm.
focus more on the fundamental basics with the curiosity. Because
Mm-hmm.
sometimes what happens during our change in the, or you can say shift in the our academics course also, there is a different kind of a syllabus they are having it. So if we
Mm-hmm.
they don't focus on the fundamental basics in depth understanding of the biotech industry, how it is working, with the
Mm-hmm.
curiosity and with lot of questions,
Mm-hmm.
then they can better adapt the biotech career. So biotech,
Mm-hmm.
as we see in that there are a huge amount of a shift in terms of a products, in terms of a technologies, in terms of a processing, in terms of a regulatory landscape. So they need to little bit more aware about the regulatory frameworks also with the compliance requirement. So that is the second
Mm-hmm.
thing that they need to focus. Third thing, that any young entrepreneur is coming into the any young you can say academician is coming to the biotech industry, they need to have a good academic versus industry partnership during their initial phase of academics. They need to have a
Mm-hmm.
good training with the industry in the industry, and in that with the industry experience and industry versus the academic collaboration, that can enhance their understanding regarding the biotech
Mm-hmm.
industry. So that is the three things I want to tell in every young every young academic
Mm-hmm.
person, that they want to
Mm-hmm.
pursue a career in the biotech industry, they need to focus on three things. If
Mm-hmm.
I will say that what they should avoid is, so they should avoid the complacency. So sometimes that we are doing the good course, academic course from the good universities like IITs, IIMs, or you can say NIPERs, and there. So, but we should not complacent about our education from the good particular academic institution, but we should focus on the how industry is also working on the some part. So we should not be complacent about only towards the academics, so that we
Mm-hmm.
should avoid it. Second, they should not too much complacent about the strategies or strategical thinking. So they should
Mm-hmm.
more focus, they should have a more focus
Mm-hmm.
on the strategic thinking also, that
Mm-hmm.
strategic thinking that can help them, that also helped me in my initial career, that how they can have a vision how they can have a decision making, and with the vision and decision making, how they can excel in the career. So that I also
Okay.
want to tell them that they need to focus on these things, and they have to avoid the complacency.
Yeah, yeah. Thank you very much for such a great tips. I think these are the very important points, especially that when they are in academic or they are working towards their academic degree, they need to focus on what is important for the industry, because there are some differences how you work in academic and how you work in the industry. So Harshit, do you have any suggestion how they can get some information about industry? I mean, can they go to some conferences, trade shows, or read about magazines? How they can learn about the industry while they are still working for their degree, or soon after they finished the degree? How they can get something about the industry?
Yeah. So one and most important things that they can have is that, see, if you see, especially in India, that there is
Mm-hmm.
not a good collaboration between academics and industry. So
Mm-hmm.
what industry and academics should focus is, especially academics should focus on the career development for the students, is that they can
Mm-hmm.
focus more and more industry driven projects, that they can send those particular students for
Mm-hmm.
those industry driven projects, so they can
Mm-hmm.
have that partnership with them to excel or to understand the
Mm-hmm.
industry aspects.
Okay.
Second, when any academics projects are there, so they may not be the 100% you can say resources to have a better understanding in terms of a scalability, better understanding about the commercialization of those projects where which is being
Mm-hmm.
handled by the students. So then they
Mm-hmm.
can also connect with the different you can say industrial persons. They can invite
Mm-hmm.
different industrial persons to help that particular projects. And other than that, there will be a that we need, they need to create some platforms where industrial experts can share their experience very easily. They can invite them,
Mm-hmm.
they can create some forums, they can create
Mm-hmm.
some channels where they can arrange some talks and the podcasts like what we are doing. So this kind of a framework we need to create between the academic industry to have a
Mm-hmm.
better understanding about the industry for the students.
Okay, okay. So that's a very important point, and the students need to be aware of these activities. If I mean, obviously,
Yes.
there should be more activities between industry and academic, but whenever there is a one, students and all the young people need to be aware of that and try to sign up as soon as possible, I guess, to secure a spot. Okay, that's a very important point. So, Harshit, one other thing which I wanted to find out is a lot of people didn't know about biotechnology and still don't know, but obviously there is a more awareness of biotechnology since the COVID times. People are aware of now what is RT-PCR, what is vaccine or recombinant vaccine or RNA vaccine. So, what do you think needs to be done to educate the public more on biotechnology? What industry should do, or what the effort should be in the process?
Yeah. So, as you mentioned very correctly, that during COVID time, people have a very good faith on the biotechnology now. That yes, definitely
Mm-hmm.
biotechnology can save their lives during
Mm-hmm.
this tough time of the COVID-19. Now,
Mm-hmm.
considering the other biotechnology aspects like the biotherapeutics and the biosimilars, the
Mm-hmm.
one common you can say mislinking things that industry versus patients basically
Mm-hmm.
are there, that patients are not aware about the scientific approaches
Mm-hmm.
what industry is following to manufacture the drug. That is the
Mm-hmm.
first thing, that we should create awareness for the patients, that how this particular drugs are being manufactured and how these drugs are being sent to the patients with how much care in terms of a
Mm-hmm.
quality assurance that we are taking.
Mm-hmm.
So that we need to
Mm-hmm.
create awareness to the patients. Now how we can aware that awareness to the patients, most of the marketing teams are going to the clinics, they will talk to the doctor, they will explain to the doctor, and
Mm-hmm.
they will try to have a good business around that particular drug. Now what
Mm-hmm.
industry should do next is that those marketing representatives, so that marketing representatives needs to be trained on
Mm-hmm.
those aspects of the scientific approaches, the quality controls, quality assurance, and
Mm-hmm.
those particular clinical trials related results, that they need to create some, you can say, audiovisual kind of a training modules that they can directly give to the doctors, and through the doctor we can create those kind of awareness to the patients, so that clinicians versus patients interactions we need to enhance with those kind of audiovisual representations.
Okay. Yeah, you are right. Marketing people and marketing function in general are very good in communicating things about their products, and they can play a very significant part when it comes to educating the general public, patients, and the doctors when it comes to biotechnology. So, Harshit, with this, now we are coming towards the end. I wanted to ask you if you could please share a funny story in your biotech career or a particular fact or surprising thing about biotechnology that you think exists in biotechnology, but general people or general public is not aware of it. So either a funny story or any fact or surprising thing about biotechnology, if you could please share.
Yeah, I will share both. So I have a funny story. So when you we used to do our earlier career, we used to do the fermentation process, or you can say bioreactor process.
Mm-hmm.
Now, what important is that our labeling practices and everything. At that time, we are not 100% aware. We used to just we are using just markers to label it. So what happened once? Then when we started our production fermentation, we have seen that instead of proper cell banking, we have introduced the other particular product cell bank. And when we got the after 15-20 days, when we have our fermentation product, or you can say harvest, basically when we purified that products, we understood that product is different. And when we seen that results, we amuse that how we can do this kind of a silly mistakes, and that can give us the different product. So then we
Mm-hmm.
understood with that funnier thing, that yes, if you don't label, if you don't do proper documentation of your things, that can impact differently in terms of your efforts, in terms of your development, in terms of your time investment. So that kind of a funny story, that how we can use different products line to produce
Mm-hmm.
the product or purify the product for our initial development process development experience. So
Yeah.
we need to learn. And now regulatory landscape is also evolving, that how important your data, so how you
Mm-hmm.
can make efficient and accurate data through different small things, like I can label it properly, I can do proper entry exits of the our things, how I can put proper labeling of the samples, that I can have a proper product without any time lapse. So that
Mm-hmm.
funny incidents can give us the learning also, gave the learning at that
Mm-hmm.
time, that we need to have a proper systematic approach even in the
Mm-hmm.
development, that can give the efficiency of process development.
Yeah, the labeling is important. There is no doubt, because once you put something in the bottle or flask or I mean any of the container, once you close the lid and you don't label it, next morning you won't remember all the solutions, all the cell lines. They will look same. I mean, it will be very tough. So thanks for sharing that. So Harshit, after this, we will do the rapid fire section. In this section, I'm going to ask you 10 different questions, and you need to respond from a scale of 1 to 10. And 10 is being best, almost there, or strongly agree, depending upon the type of question. And then we intend to finish this in about 60 seconds. So I'll start the first question. The quality of biotech drug products in India.
Yeah, 7.
7, okay. Production capacity in India.
Yeah, 6.
Innovation in India.
I will say 5.
Okay. IP protection in India.
Yeah, 7.
Okay. Adequate availability of talented workforce.
9.
Okay. Productive interaction among peers.
7.
Capital available for new projects.
8.
Okay. Cost efficiency with an aim to cut down the cost of the final product.
8.
Okay. Unambiguous and clear guidelines for regulatory process.
6.
Okay. And final question, make in India efforts.
Yeah, definitely 8.
Okay. Thank you. So with this, we end the rapid fire question. So Harshit, thank you very much for all the experience that you shared in biotechnology and how it can lead to the new things in coming 5 years, 10 years, and some of the tips for the younger generation, and all the funny stuff. And I think that's a funny, but same time it's very important. Nobody can underestimate the value of proper labeling. I mean that. I still remember my days when my supervisor told me that, Arun, everything has to be labeled. Even if it is very simple, you can clearly see and you will never make a mistake. Still, it needs to be labeled with the date and everything. So thank you very much, Harshit.
Yeah. So it's a great talk. I liked all the questions, which are very. Its framework is very complete and concise. So it's good questionnaires for me also and for other listeners also. That yes, how we should think, how we should pursue our career in a biotech, and how we should focus from our initial career to the next phase of career, or you can say mid leadership roles. So that gives me a very good enthusiasm now that I can also share a few more insights in future also in this
Hmm.
kind of a podcast. So thank you so much for that.
Thank you. Thank you.